Hi, everybody. Thank you for joining us for this seminar and for walking through campus with this great weather, great Michigan weather. My name is Veronica Berrocal, and I am an associate professor in the Department of Biostatistics. Together with Vicky Ellingrod, I am a co-lead for the work group on education and training of the Precision Health Initiative. The Precision Health Initiative was launched last January with the goal of enhancing and fostering interdisciplinary research through campus on precision health by various means, by developing tools and infrastructure to facilitate precision health through educational events, funding for interdisciplinary collaboration, and also to transform the research funding into healthcare settings. So for today, the goal is to have a presentation by Dr. Minoli Perera. So we will have a presentation and then follow up by questions and a discussion. So let me introduce Dr. Perera. Dr. Minoli Perera is an associate professor in pharmacology at Northwestern University, and her research focuses on pharmacogenomics, especially focused on African-American populations. Pharmacogenomics involves using a patient's genetic information to predict drug response, such as whether the medication will be effective or if it might lead to adverse effects. In particular, Dr. Minoli Perera's lab focuses on anticoagulants, the genomics of drug metabolism, and the pharmacogenomics of inflammatory bowel disease. Dr. Perera is an accomplished researcher. She has funding from several sources, including the National Institute of Minority Health and Health Disparities, to study genetic variation and drug responses within African-American patients. And today, she is giving a talk on African-American pharmacomics.
Hi, thank you. Can everyone hear me? All right, wonderful. So thank you so much for the invitation to speak. And the title is meant to be a little bit, I don't know if provocative is the right word, but made to be thought-provoking. So pharmacogenomics has been a field for a few decades now, and we're now understanding the need for multiple data sets and multiple what we think of as big data to really understand and uncover biology related to pharmacogenomics. So I hope I'll try and tell you a story that first is around pharmacogenomics and then kind of moves on into the omics piece of it, which is a little bit more, I'd say, translational or basic science, but much of what I do is clinical. This is a picture of the Northwestern campus. I actually work in the downtown campus, so the Evanston campus is beautiful and looks very much like a university, but the downtown campus looks like downtown Chicago. I joked that my previous presentation I gave yesterday, which was in the South, I had a picture of what the polar vortex looks like in Chicago, but as the next slide, I said it would not be appreciated in this audience, as it would just be another day in Michigan, I'm quite sure. So I want to start with just an idea of what problems we face, and to give a broader concept around minorities and pharmacogenomics. I think this is my pointer, yeah. So this was published in 2016, looking at the number of genomic studies that included populations that were not of European ancestry. And so in 2009, this blue pie piece, which is 96% were European ancestry, and everything else was everybody else, not just African Americans, everybody else. And a big push was made to add diversity to these studies. And so by 2016, the black pie piece has gotten smaller, but really, it's not a huge difference. We still don't have a very big pie piece here. And this is the breakdown by ethnicities. And I know it's too small for you to see this, but this big purple piece here is actually Asian ancestry people, which is what makes up most of the light color of that pie piece. So most of our increase has been in Asian ancestry people, which of course is very important. But down here are all the underrepresented minority populations. That's where they live: African Americans, Hispanics, Native Americans. And we've gotten a little bit better, but really not much. We still don't know very much about them. And for me, I concentrate in pharmacogenomics, and what this means is that we have lots of data in European populations about SNPs that affect drug response or adverse events, but do those accurately translate into populations like African Americans? And I would say we talk about health disparities on prevalence of disease and mortality of diseases that differ by different populations. But really, with this sort of pie chart going on, we're also perpetuating a health disparity in precision medicine that we will not make better until we have a very good understanding of these populations down here. And this is also made worse by the fact that NIH makes much of the data, genomic data we have, publicly available. But those databases are predominantly of European descent. So people like me who work on populations that are not of European descent have a very hard time, finding replication or finding other data sets that may incorporate some of or may find the same findings that I do. And you'll see in a moment what I mean. This was actually published earlier this year. Very nicely, it's a little light, sorry, showing the size of GWAS studies that have been published up to 2019, and by ethnicity, with the light blue being Europeans. And look at the size of these studies. We're now up to 1 million people in GWAS studies, much of this helped by UK Biobank's release of their data. But you can't even see these little colored dots down here. And I gotta say, all of my studies are these colored dots down here because we just don't have the data. I put this up here only for one reason. It has nothing to do with minorities. Yesterday was Women in Science Day, and I think it's important to acknowledge that most of these studies are not led by women. And so I put a little, bug in people's ears that if you are a woman, if you're mentoring a woman, if your PI is a woman, we need to have greater recognition and promotion of women, certainly in genomics. So it's just a little bit of a soapbox. I always use my presentations to make my political, unrelated to science talks. So let's go back to African Americans. And I came to pharmacogenomics actually after my PhD. It's during my postdoc, and I studied in a human genetics lab. And it was a real, a real crash course, I'd say, in human genetics, and I don't claim to be a human geneticist. But one of the things that was fascinating is the idea around African ancestry populations and why using them actually really enriches what we could learn from in genomics and in pharmacogenomics in particular. So this map is just a representation of us, our story as human species. The human species, we arose in Africa, and then through many millions of years, we migrated throughout the world in these waves of migration, these small waves of migration out of Africa. And what that means for pharmacogenomics or genomics in general is that the greatest amount of genetic diversity exists in African populations or populations of African ancestry. This quote here is actually from the Apartheid Museum. It was by Nelson Mandela, and it's one of my favorites because it's the marriage between the truth of science and the power of social justice. In a great quote that says, "Humanity was born in Africa. All people ultimately are African." And I think what I would say is to really understand human genomics, we must understand African genomics because it is really the start of us as humans. So, the oldest populations are African. They have the most genetic diversity. African Americans are admixed between Africans and Europeans. What that means is portions of their genomes come from each of these parental populations. And the mix between that can vary anywhere from 40% African ancestry to 99% African ancestry. And that's because African American as a race, and you'll note I never actually use the word race when I talk about my studies. I use the word population. Race is a social construct, is how we identify ourselves or how other people identify us. It actually is not a biological term. Ancestry is, because you can measure ancestry in a person's genome. So this variation is also a part of the African American population. There are disparities in disease like breast cancer, but there are also differences in drug metabolism. And as a person who cares about pharmacology and pharmacogenomics, this is where I find the most interest. And I use this example of CYP3A5. CYP3A5 and that family of enzymes is a drug metabolizing enzyme that metabolizes 60% of all the drugs currently on the market. go through this enzyme. That's how we clear it from our body. CYP3A5 has been known for many, many decades to have a splice variant that makes it completely inactive as an enzyme, and that the frequency of that splice variant is very high in European populations, such that they do not carry a functional enzyme. But in African populations, it's at a much, much lower frequency. So they carry a functional CYP3A5. And given the widespread importance of this enzyme in pharmacology, how does that play a role in the variability we see in drug response? And we're starting to uncover that, but I don't know that we have a full idea of how even this enzyme, this well-studied one, affects different populations. So I said African Americans have the most genetic variation, but they also have what's called differences in LD structure. So LD is caused by the fact that we inherit our chromosomes as chunks in a fashion where they're pieces of it inherited together. That means that SNPs along nearby SNPs are usually correlated with one another. And this correlation can be seen in these haplotype plots, where the more red means they're more correlated with each other. And what does that have to do with pharmacogenomics or genomics in general? Well, if you think of a hypothetical where you look at this population, and let's say the SNP5 is our associated variant. Well, really, it could be any SNP that's in red. They're all correlated to one another. But in African populations, there's much lower correlation because they're older. They've had more time for these large spans of LD to be broken up. So maybe in this population, if you look at the exact same phenotype, SNP2 may become the variant that's associated. Now, this could mean that there are two different SNPs that affect two different populations. It could mean that it actually is SNP2, and we could not tell that in Europeans, but we can in African Americans, or Africans. So that's an important piece to keep in mind as you see discrepancies between genomic studies between populations. When I started my work, I started working in the drug warfarin. It's an anticoagulant. I don't know how many people are pharmacologists here, but it's an anticoagulant. It's a very old drug. It's used across cardiovascular medicine. And people can require a dose that is 2 milligrams a day. Some people require 20 milligrams a day. And your physician will not know the dose you need, not really. He'll probably start you about 5 milligrams a day, and he'll work one way or another, titrating that dose until you're in the therapeutic range. And that's current practice, but we've known for a while that there are genetic variants that affect the amount of warfarin that you'll need. And when I was finishing my work in African Americans, these two studies were published at the end of 2013. This top study, both of them in the New England Journal, the top study is the COAG trial. It was done in the US with a diverse cohort. And the bottom is the UPAC trial, which was a European cohort. Both of these were trying to figure out if you use genotype to dose warfarin, would that improve the outcomes in patients? And they genotyped the things that they knew most about, which were the variants that were previously discovered in European and Asian populations. They exist in these three genes here, CYP2C9, VCORC1, and CYP4F2. But what we were noticing in our African populations, if you took the known highly associated variants, SNPs, in these genes and tried to explain the variation in dose in African Americans, it explained significantly less of the variability. And so some people said, well, that's because environment is just, the rest is just environment. That's all there is for genetics. Maybe, but maybe not. One of the things you notice if you're a physician treating African Americans on this drug is they, on average, require a higher dose, taking all the other non-genetic factors into account, like age or sex or renal function. Even if you account for all of those things that could affect the dose, they still required a higher dose in other populations, suggesting that there may be something else driving their dose requirement. So these are the results of the trial. In the UPAC trial, that was all Europeans. In all these kind of clinical measures of response to a drug and staying within a therapeutic range, using genotype was significantly improved the clinical outcomes. In the COAG trial, for the relatively same measures, there's no statistical difference between using genotype or just dosing at 5 milligrams and titrating. And there are lots of reasons for that, and people have debated that for a while, and I won't debate the study design of those two trials. But here's the data for the African Americans. If you just look at the African Americans in the COAG trial for the same clinical phenotypes, they actually do statistically worse if you use genotype to guide their dose. And that's an important fact, right? This was one of the things that the paper stressed. Don't use genotype to guide African American dosing because you will harm them. Well, what they're not saying in that paper is that there were known genetic variants well before my work that were specific, only found in African American populations that were not genotyped. At the same year, we ran our own GWAS in African Americans and found this SNP. This is upstream of the CYP2C9 locus. It's actually not in a gene. It's well upstream of that family of genes. It was highly associated, significantly associated with warfarin dose requirement, and it had a minor allele frequency of 25%. What that means is 47% of African Americans carry at least one copy of this SNP. And if you don't genotype them, you can imagine how many people you would be misclassifying as needing a normal dose, a standard dose of the drug, versus maybe an altered, a different dose. And we kind of quantified how much that would be. If you carry one copy of this SNP, you'd need almost a 7 milligram decrease in dose. But if you carry two, you'd need almost a 10 milligram decrease in dose. And these are very clinically significant changes in dosing. But again, that were not incorporated into the clinical trials that were previously conducted. When we were trying to publish this work, the question continued to come up. What does this SNP do? What enzyme does it affect? And we could not answer that question. And the reason is that to answer that question, you would need gene expression data, preferably in the liver if you think it's a drug metabolizing enzyme. And this SNP only affects warfarin dose in African Americans. So you'd need African American liver expression data with genotype data to answer this question, and that did not exist. So we weren't able to answer it at the time. I went on to look at one of the few disease susceptibility studies that I've done. Most of my work is in pharmacogenomics, but this was an idea out of a former postdoc in my lab who wanted to look at genetic predisposition to VTE, that's venous thromboembolism. It has higher mortality and higher frequency in African Americans. There's two kind of flavors to VTE. There's deep vein thrombosis and pulmonary embolism, which has a high mortality rate. And so what we did is we ran this GWAS. This is my postdoc. She's now faculty. And we reached genome-wide significance with this SNP on chromosome 7. But as all people in this audience are probably familiar, you always have to replicate every one of your GWAS findings. And when we went to replicate, we actually replicated these chromosome 20 hits. We were fortunate that, and this is a high prevalence SNP, 21% minor allele frequency, that in GTEx, which is the Genotype Tissue Expression Consortium data set, this was an EQTL, or a SNP, that affected the gene expression of a gene called thrombomodulin. When she told me that, I became really excited. I didn't know what thrombomodulin was, but it sounds like it should be important for coagulation. So we went on to look at a different data set of just gene expression in people with cases and controls of VTE. And this was a very significantly differentially expressed gene. So what is thrombomodulin? This is a little mock-up. This is thrombomodulin down here. It binds thrombin and prevents it from participating in the coagulation cascade. It's the last step in the coagulation cascade. And you can think of it as kind of the natural brake on coagulation. And our SNP caused decreased expression of this gene. So it makes a lot of biological sense. And we were lucky that we had EQTL data to support our findings. What we wrote at the paper was that there were very large European studies of VT at the time. The same year we published this, which is only in, I believe, 280 or something patients, a study out of Europe with 20,000 people came out, and they found what had been found previously in European populations, which was factor V Leiden and prothrombin mutations were the predominant risk factors for VTE. And when we looked in our population, there was no person in our population that carried these variants. And that's because they're exceedingly rare in African ancestry populations. And these two SNPs, they found other SNPs as well, but these two very strong associations are important because clinically, right now, if they are used as genetic predictors for VTE. So if you were, let's say, a woman who's had a VTE at a young age and you became pregnant and your physician was interested in knowing if you had a predisposition to VTE, they would run this genetic test. And if you were African American, it's more likely than not this would come back as negative. You don't have a genetic predisposition. But clearly that's not the whole story, because these are not carried by African Americans. So they cannot really predict risk. And we went on, after we published this paper, it became obvious this may actually have real consequences. So for you people who aren't in the know, this is Serena Williams, one of the top athletes in our country and probably the world. She has suffered from multiple VTEs and PEs. And one of the criticisms we got when we wrote this paper is that maybe it's all lifestyle and environmental risk for deep vein thrombosis in African Americans. She's probably one of the most well-trained athletes in the world with the top physicians in the world, and yet she's suffered multiple PEs and VTEs. So I don't know what her genomics is, but I would love to know that. And maybe it has consequences for this beautiful baby she has. The last study I want to tell you about is our newest work in bleeding. We were interested in seeing if there's predispositions for bleeding with anticoagulation. This is a major adverse event for any anticoagulation, but it's rare. Fatal bleeds are about 1 to 3%. But again, African Americans suffer from bleeding at a higher rate, and that's because some of the risk factors for bleeding are also at a higher prevalence in African Americans, such as chronic kidney disease. But there are other things: age, sex, cancer diagnosis, but really genetics had not been explored well. And this was an old study from a group in Sweden that looked at the SNPs within the genes that associate with warfarin response, if they related to bleeding. And the scale here is the INR, which is the blood measure for therapeutic, to be within the therapeutic range for warfarin. And you can see the VCOR SNPs, which are most prevalent, really don't have an effect. But if you look at the CYP2C9 variants, they have a really large effect on INR stability right here in the beginning. This is the STAR3 allele, which is very rare, again, in African Americans. So we were interested in looking at this range. The normal therapeutic range for INR is between 2 and 3. But really, the risk for for bleeding does not increase until about an INR of 4. And that's actually the definition for most of the adverse drug events for warfarin, is INRs greater than 4. So we wanted to look at actually bleeds that happened below 4. And the reason for that is, if you're thinking about something that predisposes you to bleeding on a medication, you do not want things that may cause bleeding that have nothing to do with genetics, such as you got a really bad respiratory infection and were put on antibiotics that interacted with this drug, and hence pushed your INR up very high and you bled. That has nothing to do with genetics. Or as you got older, your renal function started to deteriorate, again, pushing your INR up higher, and again, has nothing to do really with genetic predisposition for bleeding. So we wanted to enrich our population for people who might have this predisposition. And really, it was not that hard to find. Our definition were people that bled that required hospitalization. So a little bit less than what we think of as a major bleed, but still a very serious event. And we saw 66% of the bleeds we found in our group happened at INRs less than 4. But we didn't have very many of them, only 31 cases in our entire cohort. There's also bleeding risk predictors already available, though they're not really used clinically. It's a scoring scheme in which you can use these things like hypertension, bleeding, previous bleeding, stroke, use of alcohol or drugs, as points to how likely the risk of bleeding is. And the more points you have, the higher that risk is. And when we did this study, the postdoc that ran this study, she said, well, there's this schema, and I said, we should always have to, whatever we find has to do better than this, even if it's not used clinically. If we can predict all the people using this, there's no point in using genomics, because we can already do this cheaper, easier. So what we did find was a SNP that did not reach genome-wide significance, but did replicate, and in our ROC analysis, significantly increased our prediction of those with serious hospitalized bleeds while on this medication and at low INRs, INRs less than 4. And as it always is in genomics, it ends up in a gene that I was not familiar with, or is not part of coagulation in any way, but this EPHA7 gene, which is an Ephrin A7 receptor. It's most well known in neural development, as well as in cancer and cell-cell adhesion. When we looked at it in an in vitro model, our haplotype that contained our risk variants for bleeding increased the expression. So there is some evidence that EPHA4, a member of that family, a receptor family, is found in platelets, and EPHA7 is found in the endothelium. And in cancer research, there are two forms of EPHA7, a soluble and a membrane-bound form. These are tyrosine kinase receptors as well. And so the soluble form does not have tyrosine kinase activity, but it can bind and inhibit EPHA4. And it's actually been thought of as a therapeutic in cancer. So our hypothesis, which I don't have proof for, but I would love to continue working on, is that the soluble EPHA7, or maybe the membrane-bound, inhibits EPHA4, thus preventing platelet aggregation. And what I really think is important to recognize here is this is not a major coagulation pathway. We have lots of other pathways that are much stronger. And therefore, lots of people carry this variant and walk around and don't have any bleeding risk. And that's because all those other pathways are intact. But when you're on anticoagulation, and I want to say this may be any anticoagulation, you've inhibited all of those other pathways. And perhaps now this much more minor pathway plays a very critical role in preventing bleeding. So how do we find function in genomics? And this was an ongoing problem for me. So as I said, we had this SNP in warfarin. We don't know what it did because we don't have gene expression data. There are lots of liver studies in white individuals, EQTL studies. And we want to really think about the LD structure differences and the novel genetic variants and the unique genetic variants that are found in African background, genome background. So I decided to do this myself because it just wasn't getting done anywhere else. So we took African-American livers, we extracted living hepatocytes, a primary culture of hepatocytes, and we ran just a battery of tests on them because I thought if I'm going to do this, I'm going to try everything I can. So we did gene expression with RNA-seq, but we also gave them enzyme-inducing drugs, things that upregulate the CYP enzyme system, and did RNA-seq after those inductions. We did genome profiling as well as EQTL mapping, and then we gave the same cultures probe drugs. These are drugs that are metabolized by known pathways and collected curves of metabolite rate formation and parent drug disappearance so that we could figure out there's variability in that PK parameter. It's not exactly what you would see in a person, but it's correlated to what that person's metabolism would look like. And I wrote the grant in a specific way, but what we really have is interconnected types of data, drug induction. We also ended up adding DNA methylation to it, gene expression, genotype, metabolite rate formations. And you could think of doing many different types of analysis. You could do EQTL analysis. You could do looking at ancestry. This is something actually none of the other EQTL studies could do because they're not using admixed populations. As I told you, ancestry varies in African Americans. Does that have an effect on your gene expression or your methylation? Differential gene expression with exposure to drugs, does that uncover novel pathways for regulation in the liver? As well as incorporating all of these different platforms in ways that would inform us on the biology. And so what I'm going to show you is really preliminary data. It's data that we're working through right now. When I first got the RNA-seq data, it was a small set of livers. So we just did this very focused kind of candidate gene approach where we looked at PharmGKB, which is a database. If you're interested in pharmacogenomics, you should know about PharmGKB. It curates all of the associations, genetic associations to drug response. And they have a list of genes that are well established as being important for pharmacogenomics. So we looked at that list and looked at ancestry to see if any gene expression associated with ancestry in this list. And what we see is CYP2C9, which is CYP2C19, I'm sorry, which is the major enzyme that converts clopidogrel to its active form. Clopidogrel is an antiplatelet agent and is one of the big hitters in pharmacogenomics right now. So if you don't have this enzyme, you don't have active drug on board. It has lower expression with increased African ancestry. Also, vitamin D receptor has increased expression with increased ancestry. And I think that's a really interesting finding, but also makes a lot of, a logical sense. African Americans in general have lower vitamin D levels because they have darker skin. So, you could imagine that if your body has lower levels of vitamin D, maybe you upregulate the receptor to compensate for this lower vitamin D environment. And that's what we're seeing here. The green dots, I don't know if you can tell, this one and this one are both green. We were able to replicate that signal in the GTEx data set. And I want to say GTEx is a wonderful resource, but it only had 12 African American livers in which we could run this analysis. So really quite underpowered to look for replication, but we tried anyway. Once we got the full data set, we started looking at all the genes in the genome, and again, really interesting things came up. APOL1, this is a gene known to be associated with renal function in African Americans, and only African Americans. It shows very significant decreases in expression with increased ancestry. VEGF, as well as this platelet protein that's important for coagulation responses, plus many others. I'm just highlighting a few, but we found many very important genes related to disease that may help us start to understand disparities in disease. We also conducted the first African-American EQTL mapping study, and then we had to compare it to GTEx. That's now the gold standard. I want to say that we only had 60 livers. Livers from African Americans are very rare to come by, especially in their fresh state in which we can extract living hepatocytes from them. But nonetheless, we ran the analysis, and we see that a majority of our, this is a Venn diagram of our E genes, meaning genes with significant EQTLs, overlapped with our GTEx data set, but we found many unique E genes and unique EQTLs. And here's just a sampling of how these sorts of EQTLs break down. So for the things that are common here, this is a gene expression by genotype, and this is GTEx, and this is our data set. This is an EQTL in GTEx. This is an EQTL in our data set. So they match interaction, they match kind of the significant EQTL standards. Then we see some SNPs that are not at all EQTLs in GTEx, but are very significant EQTLs in our data set. And then we see a lot of SNPs that fall in this category, where this is not a significant association in GTEx because they do not have this the homozygous rare genotype, but it definitely looks like there's a trend towards decreased expression with the rare genotype. In our data set, this allele is carried at much higher frequency in African Americans. And even in a much smaller data set such as ours, we were able to see this as a significant EQTL. This is just to show you that we overlapped all of the EQTLs we found with the GWAS catalog, and we found 290 significant overlaps with the GWAS catalog that explained new biological insights into what gene was actually affecting those, not for all 290, but for some of these that were affecting that phenotype. We also looked at drug treatment, and I wanted to know if drug treatment changed the EQTL mapping landscape. And not surprisingly, baseline is this bubble here. Most of our EQTLs were also EQTLs in all the other conditions as well as baseline, so they're shared across them. However, you can see there are unique EQTLs that can be found with drug treatment, and especially with phenobarbital, had the highest change in gene expression as well as the highest number of EQTLs. That's not unsurprising. Phenobarbital hits many different, upregulates many different enzymes. But what I really thought was interesting is CYP3A5, which we actually did not find any EQTLs for, significant EQTLs in just the baseline condition. Once you put drug on board, you actually could uncover this regulatory, a potential regulatory variant within this gene. So, it's not significant at baseline, but with drug treatment, it is statistically significant in most of the drug treatments. And I thought this was interesting because as a person, you're not actually sitting at baseline with nothing on board. We're always having exposures to things, and therefore, if we're maybe not getting all of the regulation of gene expression by just measuring things at baseline. So with the last bit of time I have, I want to talk about our newest initiative, which is a center that I'm running to kind of accelerate the pace of what we're doing. So I've told you a lot about things here, where we discover SNPs that are important for phenotypes we care about. But in the real world, you do this, and then you do outcome studies, where you actually use this data, much like the clinical trials I started with, to see if there's a change in outcome for patients. And then after you see this, you actually do this implementation piece, where you figure out a way to deliver back this information to the patient, to the physician, in which they can actually act upon it to change therapy. And that's a lot more complicated than it seems. And for everything we've been doing in African Americans, we're here. But across the US, hospitals are doing this. They're implementing with screens of SNPs that predict drug response, adverse drug events, and it's all based on our current literature, and our current literature is predominantly white, as I said. So we're building these platforms that cost a lot of money and are delivering it back to patients on data that may not be applicable for minority populations in our country. So we built this consortia called ACCOUNT. This is actually, our paper just came out today describing this consortia. It's built on this idea that there's a discovering a translation piece that has to kind of churn and inform each other. And then there's lots of different pieces, from community-based projects to how do we implement outside of academia, a lot of questions that we're trying to answer. But I feel like this piece is probably the most important, which is availability of data. So I struggle with using public data. I love public data, but there is none in African Americans. And so we've committed to using anything generated out of ACCOUNT to make it publicly available through our ACCOUNT Data Commons. And as we begin to publish this data, we then release it to the community so that they can use it to inform their own discoveries. We're recruiting cohorts across 2 major cities, in Chicago and in DC. These are very clinical cohorts with clinical response. We're getting DNA. We're getting a blood transcriptome, which is the easiest one for us to get. We also started collecting PBMCs that can be converted to iPSC lines because, again, we don't actually have tissue expression. iPSCs have their own problems and their own differences with real tissue expression, but at least it's a step forward. And when I started doing this, I had a colleague who does a lot of iPSC work, and I asked him how many African Americans he could have. Could he, figure something out for me? And he said he had 0. And he has hundreds upon hundreds of iPSC lines from lots of clinical trials, and he has none that are with an African genome. So they're completely uninformative to me, or can be completely uninformative. So again, we're going to collect this data and try and convert these, reprogram these cells as we figure out what tissues may be important. We're starting with cardiovascular medications. We already talked about warfarin. NOACs are novel oral anticoagulants. They're the new drugs that are taking over for warfarin, and actually no genetic studies have been done. People say there don't need to be any genetic studies. They're not monitored. Like warfarin is. The problem is that the bleeding risks looked much better. The profile looked much better for these drugs. But now that they're out on the market, there's a lot of reports that the bleeding risk may be equal to warfarin or more than warfarin. It's hard to tell because the reporting of this is, again, a bit anecdotal. Clopidogrel, again, that's an antiplatelet agent, that we have lots of data in Europeans on genetic variants that mean that this drug will not work at all because it's a prodrug that needs to be converted. There's none of them actually look at African American populations. And we're doing many different types of phenotyping, bleeding, clotting, as well as clinical plasma measures of response. This is just a snapshot into what the translational project is doing. Again, we have to speed up the pace of delivering this back to the physician. And so this genomic prescribing system, which is developed by a colleague of mine at University of Chicago, is one way of delivering this back into the electronic health record. So you can, for each patient, they get this customized readout that's a red, yellow, or green light telling you there's genetic information on this patient. And they carry variants that mean, if it's a red light, don't they shouldn't get this drug. A yellow light, you should use caution with it. And also, if there's an alternative within the same class, and what light they have, red, green, yellow, that you could use in a different medication. For warfarin, we have this calculator that tells you if the dose needs to be lowered. And this is delivered back to the physician. They can click here and get the primary data sources for this. But quite honestly, physicians don't want to look at what the gene is and what the SNP is. They want to know that this was done on a patient and there is reliable information to help them guide their therapy. And that's what we're trying to do, give a really easily digestible form of pharmacogenomics, which is very, very complicated, back to the physician. And this is just to tell you that we have a website that profiles a lot of our community efforts, some of the products our community partners have made. We have a Twitter account. And as we release data and as we publish papers and have calls for grants, they all go up on our website as well as on our Twitter account. So I kind of talked fast. I apologize. This is just to acknowledge all of the beginning part of this presentation has been done by my lab. We have all kinds of people there. We have technicians, we have grad students, we have postdocs, we have recruiters, we have a bioinformatic analyst. I'm always hiring postdocs because, well, there's so much, there's so much data to go through. We have lots of people that help us contribute to this data, as well as lots of collaborators, only a few of them listed here across the country. And you know, I think I started a count not realizing how quickly it would grow. I thought it would be like 10 people in a room talking about pharmacogenomics in African Americans. We have an annual meeting every year, and this is the picture from the last annual meeting. It might be a little dark. I'm sitting here, and this is my co-PI, Dr. David Meltzer at University of Chicago. And every year we get more and more people coming to this meeting. It's now almost at 100 people attending. These are the major people that work in it, but not nearly all the people. We've expanded out of Chicago and DC and now have, you know, some people working in Pittsburgh, in Virginia. And we hope to grow because what we're doing is trying to encourage people to think about African American pharmacogenomics and do these studies that are urgently needed. And oh, this is the website. And with that, I will take some questions.
Excellent talk. Fascinating. For the 3 or 4 GWAS study, it's quite interesting. So when you look at the baseline expression, you didn't find any GWAS signals, but after you add inducers, you find a number of SNPs associated with the expression of the enzyme. So what's the possible mechanism behind that?
I think it's just maybe amplification of that gene expression, the expression measure. So now we have, I don't know for sure, we haven't run studies to look at what's binding at these SNPs or if there's changes by induction of increased recruitment of transcription factors or other sorts of proteins to those locations in the DNA. My guess is that we're amplifying a signal that's very hard to see with only 60 livers, so we don't have enough sample size to really see it. But when you amplify that signal with a drug inducer, maybe you can now uncover that signal. So even in small data sets, that's a useful way to uncover it. That's one hypothesis, I think, that might be the takeaway from that. I would like to make a little shout out that if there's students in the audience, I'd love to hear questions from students.
So a question about, you mentioned that there are large data sets that are primarily Caucasian population or maybe self-identified or identified by society as Caucasian, but there probably a portion of those are of mixed ancestry. Can those data be used? Can you look in there and extract meaningful information about?
Absolutely. Yes, absolutely. That's the definition of the admixed population is basically a mixed population. Even in that, we actually don't use self-identity for who we use in our studies. So even in the UK Biobank samples, they do have relatively large portions that are, in numbers, maybe about 1,000, maybe 2,000 people that with some African ancestry, meaning they have something that's not 0. And so that's useful, but because that data set is so large, when you think about any phenotype, it pulls it down again to some very small number. So the utility of a database that has 500,000 to 1 million people in it to use, and only 2,000 of them have any ancestry that's African, and then you want to ask a question about hypertension, and now you're down to a group of 200, for very complex diseases like that, you won't be able to answer the question. As I recall, UK Biobank has no drug data, so really it's useful for disease susceptibility, and again, because of the complexity of that data, it's limited.
That was a really great talk, and I just had one question. When you were talking about the soluble or membrane-bound EphA7, could you go into a little bit more detail on the expression that you saw and then what that inhibits?
So we have not run the true functional assays. So all we did for the paper was find the SNPs. The most significant SNP is actually only about 75 base pairs upstream of the gene, so right in the promoter region. And then just run a reporter assay to see if you put that haplotype in, does it change transcription? But do we know if it changes transcription in the tissue of interest, which is probably endothelial cells? Not quite yet. That's what we're working on. And so my hypothesis is that you know, if you have endothelial cells from African ancestry people that carry that SNP, they would have increased expression of CYP3A7. Nobody actually knows how that soluble versus membrane-bound piece works. There's actually no SNP, or I don't know if that's a regulatory change and not really related to a genetic variant. So my guess is there's a regulatory tissue-specific change, meaning some tissues have the soluble, some just have the membrane-bound. It's a protein that's found in many different tissues, including the brain. So there's so many questions left to answer there, right? Like, in endothelial cells, do we even have the soluble form? Nobody knows that answer. If we don't, even if it's the membrane-bound form, does it inhibit anything? We don't know that answer yet either. And the problem there is also it's a tissue-to-tissue interaction. And so it makes it a much more complex question.
Hi, thanks for the talk. So you mentioned that some people have, for the African Americans, there are some who might have, say, 40% European ancestry. And so are you looking at African Americans with West African ancestry? Or how do you, I guess, what are the parameters when you're looking at African Americans? You know, which African heritage are you looking at?
So we do what's it called, predicted ancestry percentages by West African ancestry. For the earlier studies I showed you, because in genetics and in GWAS studies, admixture and substructure have a potential confounding effect. We actually limited our cohorts, regardless of who we recruited, to people with greater than 70%, what would equate to about 70% ancestry, 70% or greater West African ancestry. In our liver studies, we didn't actually make that kind of cutoff because we were interested in that spread of ancestry. And we didn't want to eliminate, first, livers are very hard to come by, and we didn't want to eliminate any that may have slightly lower ancestry levels. Now, there is an argument to be made that West African ancestry is not the right proportion to be made. It's kind of the standard way of calculating ancestry in African Americans, but I would love somebody to come up with a much more accurate measure. That'd be great. Yeah.
Also, thank you so much. I've been engaged in studying pharmacogenomics of those who are being treated for depression, clinical depression, and especially treatment refractory depression. And I'm wondering whether you have had enough interaction. The diagram that you showed about genomic studies, we need to discover what we need to be testing. The outcome studies, let's learn if it helps. And then the implementation. In my arena, it's been my observation that there's a fairly deep-seated resistance among clinicians to sort of adopt this with the idea that it's helping them, not giving them a precise mandate as to this is what you must do, so that we can build the database, that we can essentially expand. And I'm wondering whether you've encountered that, for example, looking at any of the coagulation issues, and if so, how are you overcoming it? Because it's almost as if the field I'm in would like to say, it's not ready yet, so therefore let's not use it with anybody. And that means we also don't study it with populations that are understudied.
Gosh, that's it's a little bit of a loaded question, because I could go on about that for a very, very long time. I work with clinicians a lot. Outside of like doing all of this, I work hard to do implementation, not on a research basis, as true clinical care in our hospital system at Northwestern, which is very, very difficult. And absolutely, there's a lot of resistance. And I think there's two major things going on. Clinicians are used to clinical trials being how they prioritize the legitimacy of any sort of intervention. And if there's not a clinical trial on it, then it's not worth doing. And they feel that should be true for genetic and genomic information. And I push back on that and say there are lots of things we use in clinical medicine right now that have no clinical trial done on them, such as using creatinine clearance to dose our drugs. There has never been a clinical trial that said we should ever use that as a way to tell what a person's renal function is, or even how to dose them on a medication, but we do it without question on lots of medications. So I'm not saying we willy-nilly use genomics no matter what, but I think the standard is not calibrated correctly for this type of information. The second is that physicians think, or it's been perceived, that genetic information is some sort of prognosticator of a crystal ball, and that you'll look in it and you'll get a decision and it will be right 100% of the time. And first, we have no test like that, and if we did, we wouldn't need physicians. We just use that and never ask a person who could have error, right? But the truth is, it's one piece of information that may be completely negated by what's going on with the patient. So you find SNPs that tell you, oh, this is not the right dose of warfarin, they need to be a little bit higher. But if they're old and they're in bed and they haven't eaten for a week, well, that would be the wrong decision to make, and you would be, a really bad doctor to take what I tell you about genetics and ignore all the rest of it. And physicians do this daily with lots of information. They incorporate it in, and I think, I don't know how to fix that problem, to say this is one piece of information that is not absolute, but a piece that needs to be incorporated, that all other things considered, does it affect your decision? The other part is delivering that information at a time that matters. So if they've already given the drug to the patient, and then you tell them don't give it to them, first, they're annoyed by that, very annoyed. And second, they're like, well, we'll just see what happens, right? You don't know, you could be wrong, because genetics is not perfect. And so I think delivering it before with enough information is the only real way it's going to work. You can't do it reactively. Some things you could do reactively, but really pharmacogenomics cannot be done reactively. It must be given to them before. Yeah, I don't know that I know how I overcome dealing with their physician advocates. We are lucky that our chief of cardiology is a big advocate. He's African-American, so that's always helpful for everything I do, but also he's an advocate of genomics, so we've tried to incorporate that into our interventionalist, our interventional cardiologist for the cath lab, especially for clopidogrel. That has not flown yet, so I'll tell you a little bit about how successful I've been at that.
I'm not a student, but I learned more in the last 45 minutes that I think we can consider this a student question. So thanks for that talk. So in my role here, I try and make data available to researchers so they can study these things. I think with pharmacogenetics, what do you make for so it's very hard to tell if one, the prescription was picked up, and two, was it taken? Oh, yeah, yeah. So do you assume, for these studies, when you're looking at effectiveness of a medication, do you assume it's taken at is there a large enough group of patients that you can assume adherence?
So the easy answer is yes, we do assume they're taken. There are little tricks in recruitment that I use to try and enrich, I'll say, for people who are taking their medications. So one thing I tell my recruiters when they're recruiting patients is you ask a very innocuous question like, what time did you take that medication this morning? And if everyone will answer that question, first of all. I said, but if they hem and haw a little bit, like, yeah, I took it, I don't know, maybe 10 o'clock, like, that means they didn't take it. I said, people who take their medications regularly will know for sure what time they took it. I took it at 8 before I ate my breakfast. And I said, if they answer like that, more than likely they're taking it. That's not a guarantee. I have no guarantee. And there's no way to really track that because prescription claims information is very difficult to parse, and it's de-identified anyway. The other thing we do is clinical measures. Again, it's hard to say because if you have a low level, is it because you're genetically predisposed or other things are going on and you have a low level or you didn't take the medicine? So it is dirtiness in our data, and we try, I talk to my students all the time, phenotype is the most important thing. Actually, genomics in this is the easiest part we do. Phenotyping people is the hardest thing we do, which is getting that phenotype to be very, very precise and very clean, and that's part of it, yeah.
Out of selfishness, I'm going to ask the last question. And when we were deciding who to invite for this presentation, we decided on you because we've heard you speak before, and it's, of course, African American History Month. Right, so I'm going to ask a more community-based, popular hot issue question. What message would you like on a community level for both African Americans and researchers to hear about how the lack of this sample size and the lack of participation in these type of studies is actually impacting our health on a daily basis, and how researchers need to figure out how to better engage these communities?
So I'll start with the last thing, which is how can researchers better engage as a community? And I won't say that I've studied it or know it. I'll just state from my own. So when I started as a postdoc, I actually recruited my own patients. And I tell the story of going to my very first person I recruited. It was an African-American study. And it came in a white coat, and I said, my name is this, and I'm doing an African-American genomics study, and I want to know this, and I want to know that. And I said a lot of things that I wanted to know. And the man immediately said no. And I walked out and was like, okay, people tell me recruitment is hard, so that must be it. And then all the nurses there were actually African-American. They said, oh, you're just doing this all wrong. Please stop. And they were so useful. They were so useful in what I should be saying. And what I should be saying and what I learned, because it's actually the truth, is I should say what I care about is making these medications safer for you and your community. Because that is what I'm doing. I just was not saying it with the language that matters and that will engender trust. Now, does that always work? Absolutely not. It's very difficult. But the words you use matter so much, and your intention for what you're doing matters even more. So if your intention is, and I tell this now, I say, when you're recruiting people, you say, I only study African Americans because this is what I'm dedicating to. These are the problems I'm trying to solve. And I think that resonates more, and it means there's some sincerity in what you're doing. And so for large studies that are trying to recruit with diversity, I say diversity is always a good thing, but please remember that there is a real issue with us saying we want diversity, recruiting a large sample set that ends up being 80% white and 20% black or other, and not even black, other, everything else, and then saying, oh, that's not enough to do anything with. Let's just throw all that out. We'll look at the white people and move forward. And even for people who don't read scientific literature, they know that this is what's happening. So you need to make a concerted effort that that's not how you end up, and that when you say I want diversity, it's not just to make it look good, that it's really to power it. With our community efforts, the grants we've given for community efforts have helped a lot. They make materials, actually some of them are on our website, for use with patient populations and with physicians. And the last thing I'll end with is that I find the people that are hardest to work to convince on this are actually scientists and not patients. Patients are actually very giving for the most part. Science is not. when I publish a study and I've done a phenotype that's been done to death already in other populations, they say, well, what's the benefit? So this is only going to benefit Black people, and only 20% of them carry it. So really, it's very small in scope, and it's a mindset that would never occur to somebody when they find 1% SNP frequency in a white population that may kill them on a drug. Nobody says this is only going to affect 1% of white people on this drug. So we need to change our mindset about what's important and re-educate people to think, out of their biases. And I don't think these are bad people or racist people. I think they just have learned to think about things in one way, and it's very difficult for them to see it differently and to think it's new and original when you're doing it in a new population. So I think science education, educating current scientists, is the biggest thing I work with. Thanks.
So thank you, Minoli, for a really great talk, and thank you for participating in this very stimulating Q&A session. Our next event will be on March 13th at Palmer Commons, and we'll have Matt Solner. We will send an announcement reminding you of these events, and we would like for you to join us there.